that any creature would need to respond quickly to its environment, but certainly bacteria would. And a response that a bacteria is going to have, seeing as how bacteria are very small, any kind of response is going to be based on protein production. So their ability to respond is going to be usually something that results in a new protein being made. I think it's important that we understand like why that's the case. So when you ask why they would do that, the reason for that would be to respond quickly, and that actually could result not only in making a new protein but also shutting down protein synthesis. So you could either results in a new protein made or stopping protein production. So whether you start or stop the process, the why is something that we can easily answer. Sam. And the why is because this starting or stopping will prevent any kind of a waste of energy. Okay. So let's talk about mechanisms in which bacteria, so bacterial mechanisms, bless you, of enzyme and therefore protein regulation. And this is best taught with an example, so I want to spend some time talking about a specific type of mechanism that would be a feedback inhibition mechanism. And the best example I could come up with would be the tryptophan metabolic pathway of bacteria. So the best example of a regulated, or one good example of a regulated system of enzymes in bacteria would be this pathway. And let me show you how this works. It's a pretty simple concept. To make tryptophan, there's a series of reactions. We'll say that substrate A is converted to product B, which is converted to product C, and converted to W. And the reason I'm throwing W in there is because that's the biochemical symbol for tryptophan. Fun fact for the day. Tryptophan, W, biochemistry. Now, what is required at each of these spots here? An enzyme. Okay, so this would be like enzyme 1, and this would be probably enzyme 2, and then enzyme 3. And there could be more enzymes, but I'm keeping it simple with just 3. Now, if you turn off enzyme 1, can A be converted to B? Okay, so you stop the reaction there. If you turn off enzyme 3, then C cannot be converted to W, correct? It's a pretty simple idea. If you flip the switch at any one of these points, then the reaction would not proceed onward. Now, the other thing to realize is that each time you do this process, you do this conversion, obviously you're making an enzyme here, you're making an enzyme here, and you're making an enzyme here. So 3 separate enzymes, right? And what are enzymes most of the time in biology? They proteins, lipids, carbs, what? Proteins. And where's the information for protein synthesis? Where's that derived from? Where's that information? Where? Okay. So the way I would envision it is that you've got this DNA, it's double helix DNA. It's a poor double helix, but whatever. Right? And what bacteria will do, because they don't really have introns or exons like we do, their stuff is extremely basic, is their genes right next to each other code for specific groupings of enzymes. Good? So perhaps this right here is the gene for enzyme 1. This would be the gene for enzyme 2, and this could be perhaps the gene for enzyme 3. Do you see how their genes are back to back to back? If you're going to start with component A, whatever that may be, and if you're going to convert it to W, tryptophan, you know that you've got a series of reactions that has to occur. It would make sense that the genes would occur right next to each other to code for those individual enzymes. Right? It's like, here's a good example. I'm not a fan of Harry Potter, but recently Jamie has taken a liking to reading all the books. And those books are very long, by the way. I don't know if you've ever read those. Has anyone ever read the Harry Potter series? Okay, they're long books. Now, tell me what would make the most sense. For me, the good husband, to walk into the public library and have a public library where all the Harry Potters are right next to each other, because they're a series, and you need to have read 1 to read 2 to read 3 to read 4. Or would it make sense that I walk into a library where the Harry Potters are randomly scattered throughout the library? Do I want order or do I want randomness? Okay, so the order would make most sense here. I need all 3 of these genes to make all 3 of these enzymes, and I need them back to back to back, so I therefore would code them back to back to back. Does that make sense? Good? So once I start my process of transcription, I start reading the DNA, I could start reading it from this location right there. And I could stop once I get to this spot. That's my little stop sign. Good? So the DNA will have one start sequence, and it will go the entire way through to make all these to transcribe these individual genes, but back to back to back. And then we'll stop at this point. Right? So instead of me going to the library and just like trying to go find the first Harry Potter, what did I do? I got all 20 of them, or whatever there is. How many are there? It's like 8, right? Oh, really? Why is there 8 movies then?
They divided 7 into 2.
Oh. Huh. I bought her the last movie too. I'm such a good husband. I didn't watch it though, but I heard it was great. Yeah. So this gene right there codes for this enzyme, and this gene right there is going to code for this enzyme, and this gene for this enzyme, right? We'll have to do transcription first to make the mRNA, right? And then we'll have to do translation to take that mRNA that's made. So I'm showing you my little mRNA transcripts. Now I'll take that mRNA and translate it into a protein, which will be your enzyme. Cool? Perfect. This grouping of genes together is called an operon.